the protein kinase C PKC pathway. In this pathway, we have signaling molecule binds to the G protein coupled receptor, and we have activation of the G protein. In the case of the PKC pathway, the G protein is called GQ. Now, the beta gamma subunit of the G protein will go on to activate PLC beta. PLC beta will go on to cleave a phospholipid called phosphatidylinositol 4,5-bisphosphate, or PI45P2, essentially cleaving the phospholipid head from the rest of it. Now the rest of it is just diacylglycerol, which is the two fatty acid tails, and the glycerol. And the head part will go on to diffuse into the cytosol. At this part, the pathway splits. Diacylglycerol will localize PKC, right here, protein kinase C, to the membrane. Now this does not lead to the full activation of protein kinase C. IP3, on the other hand, and we have to remember that both diacylglycerol and IP3 are second messengers. So IP3 will go on to bind to IP3-gated calcium release channels on the ER membrane, and that will lead to an increase in calcium concentration within the cell. Calcium will go on to bind to PKC, and at this point, this is where PKC is fully activated by the pathway. And PKC, protein kinase C, is a kinase and will go on to activate downstream targets, and those downstream targets, the identity of them, will be dependent on the differentiation status of the cell, and that will lead to a response by the cell to the initial signal received. Signaling molecule will specifically bind to its G protein coupled receptor, thus activating it. An activated G protein coupled receptor will then bind and activate its trimeric G protein or G protein. The activated G protein coupled receptor binds its G protein. The G protein consists of the G alpha and G beta gamma subunit. The G protein in the PKC pathway is called GQ. That is just its name. The activated G protein coupled receptor acts as a GEF, which leads to a substantial increase in the rate by which the G alpha of GQ exchanges its GDP for GTP. The G alpha and the G beta gamma subunit dissociate from each other. The activated G beta gamma subunit binds and activates PLC beta. PLC beta, an enzyme, will hydrolyze phosphatidylinositol 4,5-bisphosphate, and we're just going to call it PIP2, producing inositol 1,4,5-triphosphate, and we're just going to call that IP3 and diacylglycerol, or DAG, DAG. So this reaction is simply this. PIP2 goes to produce IP3 plus DAG, and the enzyme is PLC beta. Okay? And this is being shown here. Here's our phospholipid, PIP2 right here, or PI45-bisphosphate, or PI45P2, and you can see that you have the head of the phospholipid right here. And this is where it's cleave off. The cleaving happens right here, the hydrolysis reaction. And the head part goes off to diffuse within the cytosol, while the two fatty acid tails with the glycerol, DAG, IP3, DAG will stay tethered to the membrane. Now both DAG and IP3 are second messengers. DAG binds protein kinase C and localizes it to the membrane. IP3 diffuses into the cytosol and binds to IP3-gated calcium channels called IP3 receptors on the ER membrane and opens them. So these IP3-gated calcium channels are called IP3 receptors. They're on the ER membrane. Remembering the ER stores calcium, so there's a high concentration of calcium within the ER lumen. The ER membrane contains IP3 receptors and ryanodine receptors, so another receptor, which are calcium channels that are activated by calcium. So these ryanodine receptors, unlike the IP3 receptors, they have binding pockets for calcium, where once calcium is bound to them, they will open and conduct calcium. IP3 binding to IP3 receptors leads to calcium release, which in turn activates and opens IP3 receptors, which leads to more calcium release. This is an example of a positive feedback loop. So in this picture, you have a mixture of ryanodine receptors and IP3 receptors, and we have the lumen of the ER on the bottom and the cytosol on the top. And you can see that calcium that's being shown with these dots here, etc., that you have a much higher concentration within the ER lumen of calcium. We know that calcium concentrations in the cytosol are kept at very low levels, at 10 to the minus 4 millimolarity, through active processes that are constantly moving calcium out of the cytosol into the ER or out of the cell. Now, when IP3 binds to the IP3 receptors, these channels will open and calcium will start to diffuse out of the lumen of the cytosol, and you start to get an increase in calcium concentration in the cytosol. But that increase in calcium concentration will lead to calcium binding and activating ryanodine receptors, which means more calcium channels are opening and more calcium is released. Now, both the IP3 receptors and the ryanodine channels have a special property, and that is, at low to medium calcium concentrations, ryanodine receptors are activated, meaning that they open. Whereas at very high concentrations of calcium, what will happen is calcium will both inactivate the IP3 receptors and the ryanodine receptors, meaning that both channels will be refractory. They'll be in an inactivated state where they cannot conduct calcium. And that leads to them, again, being in this position where they cannot conduct calcium. And what you have is sort of a calcium wave rippling across the ER membrane. That is what's being shown in the picture until all these channels are refractory. So you get a calcium wave within the cytosol. The local calcium spike will in turn inhibit and inactivate both channels, thus making them refractory for a short period of time. At low to medium calcium concentrations, ryanodine receptors are activated. At high calcium concentrations, ryanodine and IP3 receptors are inactivated, refractory for a short period of time. The local actions of calcium pumps will lead to an immediate decrease in local concentrations of calcium. And this is just noting that the process to remove calcium from the cytosol is always active. So once these receptors, IP3 and ryanodine receptors, are in this refractory state, then the only thing available is these off switches, which will lead to calcium concentrations within the cytosol crashing immediately. The actions of these two channels in the ER membrane leads to a short-lived calcium wave in the cell. So in this picture, the orange, yellow, red is calcium. Let's read the legend here. This starfish egg was injected with calcium-sensitive fluorescent dye before it was fertilized. So that's the dye that's coming up, kind of a reddish, orange, yellow color. The more reddish it is, the higher the concentration of calcium. The yellow color means low concentration, yellow and green. A wave of cytosolic calcium, red, released from the ER, sweeps across the egg from the site of sperm entry, the arrow. So sperm entry is right here. This calcium wave changes the egg surface, preventing the entry of other sperms, and it will also initiate embryonic development. The initial increase in calcium is thought to be caused by a sperm-specific form of PLC that the sperm brings into the egg cytoplasm when it fuses with the egg. The PLC cleaves PI45P2, PIP2 here, to produce IP3, which release calcium from the egg ER. The IP3 activation of IP3 receptors and ryanodine receptors, which leads to creation of a short-lived cytosolic calcium spike, can be shown as follows. So let's write this out. So we're going to pick up the pathway after IP3 release. So we have IP3, which goes on to activate IP3 receptors. Remember, the activation for this is these channels opening and conducting calcium. And this is on the ER membrane. This will lead to calcium release in the cytosol, meaning that you're increasing the calcium concentration in the cytosol. And of course, once you start releasing calcium, you go ahead and activate the ryanodine receptors. And these are also on the ER membrane. These are calcium channels, meaning that you're going to increase the amount of calcium, so this is a positive feedback loop. Calcium, as its concentrations increase in the cytosol, will go on to inhibit both the ryanodine receptors and the IP3 receptors. Calcium binds to the DAG-bound PKC and activates it. PKC then activates effector proteins. The response elicited by activation of the PKC pathway is dependent on the cell type. So here we have another table showing the activation of the PKC pathway and what type of responses you can get from different cells, and also showing you the signaling molecules, and here are the responses, and here are the tissue types, or the cell types. So in the liver, the signaling molecule is vasopressin, and you get glycogen breakdown. In the pancreas, acetylcholine is a signaling molecule, and that leads to amylase secretion. Smooth muscle, acetylcholine, will lead to muscle contraction. Blood platelets, thrombin, platelet aggregation. The PKC pathway can be written as follows. We have the signaling molecule, which goes on to activate the G-protein coupled receptor, which goes on to activate GQ. GQ goes on to activate PLC beta, which will lead to 2-second messengers being produced, IP3, and DAG. DAG will go on to localize PKC to the cell membrane. Now what you have is IP3 will go on to activate IP3 receptors, these are on the ER membrane. This will lead to calcium release, increasing intracellular or cytosolic concentrations of calcium, which will lead to activation of ryanodine receptors. Of course, this is a positive feedback loop, meaning that you're going to get more calcium release, but calcium will go on to inhibit both the ryanodine receptors and the IP3 receptors. But calcium will also go on to fully activate PKC. Now, at this point, PKC will go on to activate proteins that are downstream, and that will eventually lead to a specific response depending on what proteins are activated by PKC. PKC is a kinase. It can go on to phosphorylate downstream targets. Those targets will be different depending on the cell type because of differentiation. Okay, and we'll stop here and continue with the rest of this section later. Thank you, guys.